Hello everyone, this is Andy from Med School EU, and today we're going to continue on with molecular genetics. More specifically, we're going to discuss the process of DNA replication. So here we have three different replication models. So this way we could tell that there's three different ways that we could replicate something. However, in terms of DNA, it's always going to be replicated in terms of the semi-conservative model. And in this experiment, they used, I believe, nitrogen iodine or some sort of nitrogen radioisotope. And what they've done is they proved that DNA replication occurs with the semi-conservative model. What we have is our parental strand. It's of course double helix, and with the semi-conservative model, each strand are going to be separated. So they're both going to be separated by specific enzymes, and then each strand is going to be replicated on its own. So here we have one side, so one strand is being replicated and the blue new strand is being added. Same thing happens on the other one, and we typically end up with two DNA molecules, double-stranded. One strand of each has the parental strand, and one is the new strand. So that's the semi-conservative model that we follow with our DNA replication. However, there's two other ones that exist. There's conservative model, where the DNA of the parental is just copied the same. So you produce a double strand of the new, and you still have the double strand of the parental. And the same thing, as you can see, continues to occur. And there's also the dispersive model, which is completely random in terms of which nucleotides are replicated and which ones are not. So it's dispersive as it occurs on different parts of the DNA double helix. So now we're going to discuss DNA replication initiation. So in the previous lecture, we talked about how DNA is a double helix. It's got these nucleotides. They're paired ATCG. That's the complementary base pairing. And how are they going to be replicated? And more specifically in this slide, we'll talk about the initiation, how this whole thing begins. Now, this one is more specifically for prokaryotes. However, in eukaryotes, the mechanism is very similar. So what happens first is that a helicase is going to bind to origins of replication. So we've got something called origin of replication. And helicase, so helicase is the enzyme that's right here, right? It's this one. And helicase is going to bind to it, and it's going to break off the hydrogen bonds between the nucleotides and create a replication bubble. So it's going to be replication bubble. And we're going to see this in the next couple of slides, where the helicase is going to keep going. If you know how you would use a zipper, it's just going to split off the two ends, and it's going to create kind of like a replication bubble, because these things will go in both directions. So replication bubble in both directions. So I guess a good analogy to use is if you would take, if you would undo a zipper, but you would begin at the very middle, so both sides are zipped up, and you just begin in the middle and you pull it down, it's going to create a bubble going upward and a bubble going downward. And this is typically how DNA replication is initiated with helicase. Now, the significance of DNA gyrase is it prevents coiling. So because we are doing, and we know that DNA is a helix structure, so it's going to be wound around in this sort of arrangement. And if you are going to be, if you're going to have helicase break off those bonds, it's going to coil even more, right? So it's going to have these supercoils And you don't want supercoils because then DNA could get damaged. So what we have is a DNA gyrase, which is a form of topoisomerase. And typically these simply, their function is to simply prevent the coiling of DNA. Now, the helix, the DNA helicase, is going to only function with the breakdown of ATP. So we know that DNA replication requires energy, requires hydrolysis of ATP. And that's going to be important because a lot of questions come up asking they ask for the knowledge of whether or not ATP is used in certain systems and certain mechanisms. Well, in DNA replication and generally in all kinds of RNA transcription and DNA transcription and translation, ATP is used in all of those because this requires quite a lot of energy in order to perform the function. Next thing that occurs after the DNA double helix has been separated into single strands, we have the laying down of primers. So this right here, if it was separated, we have an enzyme called primase. Primase is going to lay down primers. So this is a primer. And primer is simply just a sequence of nucleotides that corresponds to the DNA sequence. And now, because there's a primer, we can have another enzyme called DNA polymerase. So we're going to label it, let's say it's kind of an enzyme like this. And this DNA polymerase. So DNA polymerase always works in 5' to 3' direction. And now that it's attached to the primer, the DNA polymerase is going to lay down nucleotides and attach those to the existing primer. And it's going to continue in this strand doing the replication in the 5' to 3' direction. However, there are several other nuances that we must mention because it is a double helix and that creates a little bit of confusion. So let's talk about more specifically about the DNA polymerase action. We mentioned earlier that this is the 5' in our previous video, right? So this is the 5' down to the 3', and that's how the growth of the new strand is made. So this is the growing strand, and this is the template strand that is 3' to 5'. Now what's important to notice is that the DNA polymerase is going to use deoxynucleoside triphosphate. So remember, nucleoside is the 5-carbon sugar plus the nitrogenous base. And this is called a deoxynucleoside because it doesn't have the hydroxyl group on the 2', it just has the hydrogen group. And instead of having just the one phosphate as it typically would, it has a triphosphate. So there's 3 phosphates, and this is the simple molecule that DNA polymerase takes, and is going to add a corresponding nitrogenous base. So of course, if this is adenine, we must have a thymine. So a thymine is incoming. Now what happens is that it uses the alpha phosphate, and the alpha phosphate is this one right here. So we could circle that. That's the alpha phosphate. And this alpha phosphate is able to make a phosphoester bond. Remember, we talked about phosphoester bond with the 3' hydroxyl group. So this is the free 3' hydroxyl group, and of course the phosphate exists on the 5'. So as you can see here, there's 3 phosphates: alpha, beta, gamma. And alpha phosphate is the one that's going to be used, and the diphosphate is going to be released. And so the alpha phosphate is going to be used to make a phosphodiester bond. So phosphodiester bond. And now this NTP nucleotide is attached to the 3' end, and now the new 3' end becomes right over here on the OH, and that's the new 3' end. And then of course it takes to the next DNTP and it binds it corresponding to the template DNA strand, and that's how they're going to build on a molecular level. So now that we're done with replication initiation, now let's talk about replication elongation. So we have, of course, our replication bubble, because the helicase is going to be opening it up. And of course, we're going to have replication going on both strands at the same time, and they're going to be going in both directions. So here, if we zoom in right there, we must notice that the replication does not happen in the same direction. It occurs still 5' to 3' because that is just how DNA polymerases work. However, there are leading strands and there are lagging strands. So there's strands that are going to lead and there's strands that are going to be going behind. So if we're zooming in on this side of the replication fork or the replication bubble, what we have is our 5' to 3', and it's going in a smooth, same, one directional without any interruptions. This is called the leading strand. Leading strand. And of course, the yellow is the parental strand, and the red and the bluish is going to be the new strand that's forming on the DNA. So this red one here is the leading strand, and these ones right here, because they have these cuts and these sections there, they're going to be the lagging strand. So lagging strand. Now, why do they have these weird cuts and stops? Why is it not continuous? Well, because the DNA polymerase must only work in the 5' to 3' direction. So it must only work in this one direction. And you cannot bind to somewhere that is non-existent, right? So the DNA polymerase would have to bind right here for it to go 5' to 3' all the way down without any interruptions. It can do that on this side because that is where the replication fork has opened, and that's the direction it's going to continue to open. However, you can't do it on this side because this structure just simply hasn't opened yet. So what they do is they form these little fragments. They're going to be called Okazaki fragments. Okazaki fragments. And we will see in more detail how those Okazaki fragments are made. So now if we're taking a look at the other side of the fork, of course, we're going to have the 5' to 3' leading strand going on the other side now. And we will have the Okazaki fragments going on this side. And it's just going to continue as the DNA is going to unwind with the helicase, and DNA polymerase action will always continue in these directions. However, here it's going to create nicks in the DNA, and we're going to see how those nicks will be resealed. So here, what we have is our, of course, it's easy on the 3' to the 5', right? So this would be 5' right here. On the 3' to the 5', we're going to have our DNA laid down 5' to 3' direction, and we're just going to use DNA polymerase 3. So DNA polymerase 3 is the main DNA polymerase that is used in prokaryotes in order to do their replication in this 5' to 3' direction. So they're simply just going to attach to the RNA primer. Remember, primers are laid down by another DNA enzyme called primase. So primase will lay down this primer at the very beginning of the 3' origin of replication. Now the DNA polymerase 3 will bind to it, and it's simple. It's just going to continue going while the helicase is going to be unwinding these. It's very simple that way. However, what happens is we have to remove that primer because it's an RNA primer, right? And this RNA primer cannot be in our DNA double helix that's going to be made because RNA contain other nitrogenous bases. So they contain uracil instead of thymine. So no thymine, but they have uracil. And that doesn't slide. We cannot use that in DNA molecules. So then we get our DNA polymerase 1 right here to do its action and do its 5'. So this is called a 5' to 3' exonuclease activity. This is the only DNA polymerase that does 5' to 3' exonuclease activity. An exonuclease meaning that is going to remove RNA primers in the 5' to 3' direction. And exo meaning it's going to begin at the beginning of the molecule and finish at the end. Endo meaning it's going to begin at the middle or the center of the molecule. Now, in this case, of course, we want to remove the whole primer and lay on a new DNA sequence. So that's going to be done by this DNA polymerase 1. It's also going to be used on the other side with the Okazaki fragments. So as you can see here, we have the primer. Okazaki fragments has been finished. And now it's going to be removed by the DNA polymerase 1. Now, once the DNA polymerase 3 has hit the end of its nucleotide, and this last nucleotide that is close to the RNA primer, now it's going to dissociate. It forms its Okazaki fragment. And now the second RNA primer must be removed. Now the DNA polymerase is going to migrate to the next part and make another Okazaki fragment. And it's just going to keep going in this direction. While this one is easy, it's just going to head over DNA polymerase 3 and it's just going to keep going in that sort of direction. Now another thing I wanted to point out is these little structures right here. Right? So these are single strand binding DNA binding proteins. So single strand DNA proteins. And the significance of these single strand DNA proteins is they protect the DNA from exonucleases so that some random enzymes do not come and cut up the DNA and make a bunch of mistakes in our genome. And another thing that's really important is they prevent re-annealing because these complementary base pairing are extremely attracted to each other with the hydrogen bonds. So even though helicase is going to break them apart here, they are simply just going to rewind back as soon as the helicase has passed to that point. So these single-stranded DNA proteins are going to bind to the DNA that has recently been opened up by helicase, and they're going to prevent them from re-annealing. So now that we have reached replication termination, it occurs differently in prokaryotes and eukaryotes. However, since our syllabus does not mention prokaryotic replication, we're simply going to talk about how termination occurs in eukaryotes. And it's quite simple. It's a lot simpler than in prokaryotes, because prokaryotes have typically circular DNA. They have their plastids are circular DNA, so they're going to have a different mechanism for termination. However, in terms of our molecules of eukaryotic termination, they're going to undergo something that's a much simpler process. So here we have our replication bubble. Now another thing to notice is there are multiple origins of replication called ORI. Wow, that's hard to say. ORI, origin of replication. And origin of replications, there's multiple in eukaryotes. However, prokaryotes only have the one. They just have one origin of replication, and then it continues on throughout. However, here, you see here we have one, we have two, and they're going to continue until they meet each other by joining together. And here we can show this, how termination really occurs, is that once these strands really meet each other, the DNA polymerases dissociate, so the bubbles are gone, because now we have two strands of DNA double helix, and the DNA polymerases will simply bud off, and they will lose their connection with the template, because there's going to be no more template available to replicate since everything has been replicated. So this mechanism of replication is extremely easy. Now, what's another important thing is that each of the new strands, each strand, and especially around the Okazaki fragments, they're going to have these nicks in the DNA. They're called nicks in DNA because the removal of RNA primer is not perfect. So what we're going to have is another DNA enzyme called DNA ligase, and is going to use ATP again. ATP. And this DNA ligase is going to reanneal the nicks. It's going to reseal all of these nicks, and it's going to fix it and make everything into a nice straight line here. So once these DNA nicks are sealed, and they're sealed quite quickly before replication termination has been reached. And then once the bubbles have disintegrated, because all of the DNA has been replicated, the DNA polymerases will bud off. They will dissociate from the DNA template, and replication has now been complete. So this concludes our lecture on replication of DNA. And in the next one, we're going to take a look at protein synthesis. And because it is a long topic, we're going to talk about transcription first, and we're going to break this protein synthesis down into two sections. And that first section is going to be transcription.